It is Thursday, July 16th, and you are watching the BioTek Voyager. It's the place that covers what's next in biotech. We're mapping the early stage
signals. It's the Jeff Show. Ben's muted himself. We are experts here, people.
Can you hear me?
Yes.
Okay, there's some sort of like weird feedback that's coming through.
Not on my end.
Just a second, Jeff. Take it away. I'll be right back.
Yeah, we will figure out Ben's technical difficulties with your camera and your microphone. Anyways, I can start by going through the leaderboard here. We're going to cover a couple of these companies today, but not all of them. The top one, right. As for those of you who don't know, as news comes in, we are taking those pieces of news and categorizing them and characterizing them and giving them each a score so that you can quickly and easily go on to the BioTechVoyager.com and find all of the early stage companies and figure out which ones are like heating up right now, which ones are hot. If you go to the leaderboard here on the homepage, I always like to kind of click through the topics that I'm most interested in. Like, let's just look at like cell therapy. Here's like the top early stage, quote unquote, cell therapy companies. And speaking of one, Co-Regen is one of the companies we're going to have on the show today.
That is correct. I figured out the problem, Jeff.
That. What is the problem?
I'd opened up the YouTube stream as well, so it started playing that at the same time. So I was hearing myself twice. Mm-hmm. We're good to go now. Excellent. Well, yes, folks, thank you for coming. Let's just get started here with the first banner of the day, which is, of course, the early stage leaderboard review. If you saw my post on LinkedIn this morning, you may have already seen this, but there are some pretty cool companies in this list. Alchemab, right, Jeff, was in the top spot a couple days ago, but Avir Therapeutics has definitely taken its spot. But I'm still really impressed by Alchemab. Like, what a cool company.
Yeah, what did they call people? They called them exceptionally resilient or especially resilient individuals.
Exceptionally resilient.
People who tend to not get diseases, and they're just like, we're going to study your blood. Thank you.
Yeah. We're going to make therapeutics out of that. Very much. So, cool.
Yeah.
Excellent.
But anyway, so if you want to like jump into the first story, I'm actually really excited to kind of get into Neutrolis.
Yeah. We can. I guess just quickly, we have two different guests that are going to be coming on the show later today, so stick around for that. We're going to be having the Chief Medical Officer of Co-Regen at 11:30, and then the CEO of Axiom at 12 noon EST. Both of them working inside of the cell therapy space. So really excited for that. One in the cancer space, one in kind of the more like the neurological damage space, I believe. So it's going to be a cell therapy focused one from the perspective of the guests, but overall we can get to some of the key signals that go beyond that narrow scope to the rest of biotech.
Mm-hmm. Yeah, so this company that I want to talk about today is called Neutrolis, and they're developing therapeutics that are meant to degrade nets. And they have use cases in autoimmune disorders and also I think maybe some in oncology. But Ben, do you know what a net is?
I've never heard
in terms of like immunology.
No.
So
I could guess, but please educate me.
Yeah, so it's a neutrophil extracellular trap. Okay, and
Interesting.
basically what these things are is they're like neutrophils will like spit them out, and they are bad. They cause inflammation. They can actually like in autoimmune disorders they can actually I think it's what like you can get autoantibodies against them, and it kind of causes a lot of inflammation and stuff. These nets, and then there's some evidence in cancer where the nets can actually protect the tumor from drugs and protect like circulating tumor cells. They can kind of encapsulate the cells, and they're actually kind of they're thought to play a role in metastasis and stuff. These nets, and there are some companies out there who are developing degraders, like net degraders. And in particular, Neutrolis is developing a DNase 1L3 fusion protein that is going to find and degrade these nets, and they use it for autoimmune disorders like systemic lupus and rheumatoid arthritis. So it's a really interesting piece of biology that not many people think about, and specifically from neutrophils, right? And the recent thing that happened with them is they published an NEJM. the New England Journal of Medicine, which is a pretty big deal. And I guess one of the bigger,
we may or may not have covered another article from the New England Journal of Medicine earlier this week. A lot of cool applications coming out of that. Yeah.
So I don't really know. Like, apparently, this compassionate use, like getting compassionate use data in NEJM is kind of a big deal. I wanted to pop open this press release really quickly to see, like, so the results provide first human proof of concept that enzymatic net degradation may address a key driver of disease activity in lupus, with broader implications for other net-driven autoimmune and inflammatory diseases. So, forgive my ignorance, Ben. Is compassionate use like when patients don't really have anything else to take, they can just kind of be given this as a last resort?
Basically. And again, if anyone else has any further thoughts on this, feel free to drop a comment and we'll get to it. But yes, my understanding of compassionate use is very much like, hey, you've got something that's really bad. There's no other options. It's like, can we please just kind of throw something at the wall and see if it sticks? And it's basically giving you emergency authorization for like an IND of 1, is my understanding.
Right. So check this out. Clinical symptoms resolved within 6 hours of the first infusion of NTR 441. A transient recurrence of rash and episcleritis occurred within 24 hours. Okay. That's crazy. 6 hours?
That's incredible.
Like, yeah. So again, I'm guessing that these nets are like actively causing inflammation, and you can degrade them and that inflammation basically goes away immediately. That's really intense.
So what's the normal use of these nets, or are they just kind of like aberrant structures that get made that are kind of like a messed up situation?
That's a really good question. I know. Like, let's just like show you a picture of them, because they are meant. I think they're meant to like capture bacteria and stuff. Like, it's a part of the innate immune response, right? And they're really meant as, I'm almost positive, like they're meant to like, these cells can just kind of puke them out, right? And it's meant to be a mechanism to like trap and kill bacteria. They're called traps for a reason, right? So you can see here they're full of all kinds of stuff, and it's DNA. I'm pretty sure that there's most of like the netosis processes actually kill the neutrophils, right? So they're just going to die. They kind of like kamikaze bomb the bacteria or whatever. So yeah, they're used like as, it's a good piece of our innate immune response. But in cancer and in autoimmune disorders, they're not. They're very nasty. Okay. And basically what Neutrolis has done is they developed this soluble circulating able DNA enzyme that will go in here and just kind of degrade these net structures. And apparently it can resolve symptoms within 6 hours of administration, which is crazy to me.
That's so impressive. Okay, like aside from that, this is something that I think this is something we've talked about recurrently, Jeff, right? I'm a big fan, and like compassionate use is one of these where it's like, hey, we've got someone who's really sick. Can we please give someone license to go and try something new? They are kind of taking on all of that risk. Eventually, every once in a while, it's not going to work. But let's try to figure out some of these new innovative medicines on people who are willing to take that risk on, and let them go and do it, so long as they kind of play within some certain guidelines. And that should really just force the field forward in such a much more meaningful way.
Let me think as to why that would be stupid. Let me think. I don't know. Let me think. I'm having a hard time. Yeah, I don't know. Like, is that like not okay to just be like, hey, we don't know how it's going to work. We don't know what it's going to do to you, but like, it might help. So here you go. Is that like taking advantage of undereducated patient populations? Possibly?
No. So my understanding is, and I could get this wrong, but like it is pretty much the patient themselves, or like along with their doctor, that are initiating this IND trial. So it's not opportunistic the way you might think it is.
Okay. Yeah, I'm gonna guess like there's a term for it. It's a compassionate use, right? So it's obviously like done in the clinic, right? I don't know, right? Like why wouldn't you do that? Although I could see companies not wanting their therapeutic to be used as compassionate use because it might give like false negatives. Sure. You know? Like here's our new therapeutic. We're going to test it in this compassionate use case in like beaten and battered patients who are on death's doorstep. And oh, what do you know? It never works. And it's like, well, is it because you're using it like at the end of the line? Like could it be that confounding factor as well? I don't know. I'm just kind of speculating here.
Yeah.
Right.
But regardless, 6 hours later, clinical symptoms are gone due
Yeah, that's
to
absolutely
degrading
crazy.
the nets. Wild.
Right. Yeah, and I'm wondering. A part of me wonders like what other pathologies are these nets actually implicated in? Like, I know when I was looking into this company, one of the things that blew me away was the role of nets in cancer, and particularly in metastasis. So if you have a primary tumor like in your lung or in your liver, right? The neutrophil, the first kind of immune cells that get to a tumor are neutrophils and macrophages, typically, and then like fibroblasts. They cause all kinds of like wound healing processes. The neutrophils can spit out these nets, and they actually like form like barriers around clumps of cancer cells. And then if that clump of cancer cells escapes the tumor microenvironment and is circulating in the blood, immune cells in the blood can't find those cells because they're encapsulated by nets. It's like armor for the cancer cells. And then there's also evidence that suggests that the nets can actually create like a landing zone in other like metastatic, locations. And that's kind of where the metastatic diseases kind of take hold. So degrading the nets in autoimmunity, obviously, it can resolve symptoms within 6 hours, apparently. But in cancer, there may actually be implications in like preventing metastasis and stuff, which would be insane. And also at the primary tumor sites, degrading the nets can allow for increased infiltration of immune cells and even increased diffusion of drug compounds. So there actually is a lot of potential in this seemingly obscure region of biology. Like, who would have thought of like neutrophil nets as an interesting thing, right?
Interesting. Yeah, that's a really good point as well, right? Like maybe this is something that's also contributing to metastasis and going to other places, right? Because you've basically got like Velcro on the outside that's really good at grabbing small stuff that would try to get at you to harm you, but it's super good for like grabbing onto something else later. And so if we can degrade that, it's a win-win all around. Cool. Mm-hmm. Excellent. All right. And the next signal of the day is Novu, Jeff. This one is mine, but Novu is really cool.
I can't spell it. Oh, there it is.
Novu. It is French.
Got it. Yeah.
Novu means new. So it's
I'm sorry. They're from Scarsdale, New York.
Well, maybe some of their leadership spends time in Europe.
Oui, oui.
Oui. But yeah, in particular for this one, Jeff, the overarching interesting thing here, and there's a couple different things I want to get into here. One of them, first of all, they're led by a gentleman, I believe if you just scroll down a little bit, a guy has like a huge track record, Owen A. O'Connor. He's a giant inside of the field, and so he has a very successful track record of kind of going through and making some really successful cancer therapies. But in particular, what was super interesting about this approach is they are basically taking existing approved oncology drugs that have really high toxicities. Mm-hmm. Right. So, you've got some sort of small molecule, you can use it for chemotherapy or something else that's going to be, equivalent to chemotherapy, kind of blasts the cancer cells, but blasts everything else. Yep. Not super good for you, but when you're on death's door, it's better than nothing. They are able to take these approved small molecule drugs that have high toxicity and they stick them inside of a nanoparticle. Mm-hmm. And so basically you're able to shield them from the rest of the body, everything else that's healthy. You are able to preferentially get it into the tumor microenvironment or to the cancer tumor, and then it uncoats and it can be as toxic as it wants to be, now that you've gotten it precisely to where it needs to go. So it's actually pretty elegant. So I like it.
I'm going to test your biology connecting the dots ification knowledge.
Oh boy.
Okay. So if they're using, we'll call it a lipid nanoparticle, some kind of like nanoparticle, right, to deliver therapeutic drugs to target cells. If they are using it for cutaneous T-cell lymphoma, what technology is this akin to? And I'll give you a hint.
A cream.
I hate this technology. Or I am known to publicly talk against this technology.
It's an in vivo approach.
Exactly. It's essentially take the in vivo CAR T idea, and instead of putting a CAR encoding DNA construct, you just put drugs inside of it. So this nanoparticle will deliver the payload to T cells, but in this case, the payload is not a CAR construct. It's just some kind of cytotoxic chemotherapy or what have you, right? And we know from the in vivo CAR T cell space that we can get like CD8 or T cell specific delivery of the constructs. So instead, like if we can do that for in vivo CAR T cells, let's just treat T cell lymphomas in the exact same way.
Nice. It looks really promising. And again, the fact that you're using an existing chemotherapy or cancer therapy drug that's already approved, just has high toxicity, and then sticking it inside of a nanoparticle looks pretty cool.
Yeah, so I'm wondering, like, I want to know, chromostat seems to be like their lead candidate, but what is the actual like therapeutic compound? I bet you it's just like etoposide or something. You wouldn't need it to be very specific. Probably. It could be just like poison. Anyways.
Yeah. Well, regardless, folks, we're going to be having 2 interviews today. One at 11:30. The very first one is going to be with the Chief Medical Officer of CoRegen, and then at 12 noon, we will be discussing an additional cell therapy with the CEO of Axiom Biosciences. So stick around for those. The first one will be coming up in around 10 minutes. Dr. Sunil Gupta has already joined the green room, so we're locked and loaded with our first interviewer in just a few minutes here. We'll get started.
We got it.
But in the meantime, we'll try to get through some of the key signals from the day.
Yeah, this is really cool, man. I like this. I don't know why. Like, this is an area where we should start doing, dude. We should start doing things where like we pick a type of technology and go through the companies who are kind of in that associated technology. This is like innovative drug delivery mechanisms or whatever, right? There's probably a bunch of companies in the BioTek Voyager who are doing something similar. It'd be really cool to have like a dedicated section of like, hey, we're going to talk about companies who are doing this today.
Yes. That would actually be a really interesting like recurring event, right? It's like, hey, once a month we are doing something dedicated to, what have you right. Technology, and all of the companies that we're going to be covering, and ideally we are able to even to get some of the
bring on some people.
You bring on some people as well. That would be super cool, Jeff. Yes. That could be something that's kind of going on in the background for later folks, which is our plan to kind of start doing like one-off events with the BioTek Voyager above and beyond our regularly scheduled programming. So
yes,
I like it.
I wonder if I have like drug delivery as a thing here. I don't think I do. Oh, advanced. Let's see, advanced delivery. I wonder if continue speaking, but I want to know like if what's it called?
Novu
is on here. I bet you it is. It has to be, right? I'm not good at the alphabet.
There we go. N-O-U. There you go.
There it is.
Novu.
There it is.
Perfect.
Yeah. Advanced delivery. See, we could easily do this.
That'd be phenomenal. Let's do it.
Cool.
Right on.
Cool. Who's next?
Who's next? Jeff, I believe this one is yours. It is Senti Biosciences.
Ooh. Ben, you know how much I love NK cells. You know how much I love NK cells.
NK cells.
You also know how much I love cool biology.
Yes.
So imagine my joy when you have a NK cell company with cool biology, which is precisely what Senti Biosciences is. We talked about Senti Biosciences a long time ago when the BioTek Voyager just started. They are doing logic-gated CAR T cells. Or, sorry.
Whoa,
I've betrayed my beauty.
Hold up.
Logic-gated CAR NK cells. Ben, I have recently kind of become like a computer nerdy, coder nerdy dude.
Sure.
So the idea of like logic gates and all these different things, it kind of comes from software, right? Senti and a couple other companies have built logic-gated CAR constructs. You know what logic-gated CAR constructs are, correct?
Of course, we have discussed this before.
Okay, so let's see. Senti Biosciences is one of
the. But let's refresh for the audience.
So yeah, what they are is essentially like you can have a a circuit, like a car circuit built into your car approach, I guess you could call it. So normally a CAR T cell is one CAR construct, and it's like a positive activator, right? So you can say, okay, if my CAR construct binds to antigen 1, kill cell that expresses antigen 1, just 1-to-1 positive signal.
Sure.
However, what if that antigen is highly expressed on other tissues, right? But also on your cancer cells, right? Now you'd be killing all kinds of healthy tissues. So what if you took another CAR construct and said, okay, if we have CAR number 1 binding to antigen number 1, that's positive signal. However, typically our cancer loses the expression of something else, but the healthy cells will continue to express that thing. So we have another CAR that will bind to the healthy cell antigen, and it's negative. Right? So you have one CAR that's positive and one CAR that's negative, right? So you want the cells that have antigen 1 but not antigen 2 to be killed, right? That would be a and not logic gate.
For
antigen
sure.
1 and not antigen 2.
Yes.
And you can imagine you have like an and gate where it's like you need to have antigen 1 and antigen 2 present. Don't kill cells with antigen 1 or antigen 2. That would be an and logic gate.
Right.
Senti Biosciences has a lot of different gene circuits that they build into their CAR T cells. It's actually a really interesting area of biology. And I will say, anybody listening, shout out to my friend Everin over at Karolinska. Look into the company Vycellix, Vycellix. They're going nuts with gene circuits. Super, super interesting. They have our CAR constructs can release a TF that causes cytokine expression and all kinds of crazy stuff. But Senti Biosciences is kind of one of the pioneers in the area. If you were to say, like, give me one company that's doing logic-gated CAR constructs, I'd be like, well, Senti is like the company that's doing it. So they recently, Senti Biosciences spins off Senti 202 gene circuit assets into private new company, pivots to regulator dial platform. Okay, so new company alert. New company is going to come online with logic-gated CAR T cells or CAR NK cells coming from the Senti Biosciences group. Really cool for us. Ben,
new company alert.
Yeah, new company. Okay, sure. that can be a thing now. New company alert. Just record that. Use that as a soundbite.
No, we're not doing that.
Stick to what you're good at, which is nothing.
Yeah.
So yeah, pretty cool.
So yeah, the question becomes, what have they seen that kind of causes this to be why they feel the need to make this into a new company?
I'm gonna guess it's because here's what I'm noticing with these companies like this. They start with an initial idea, right? And that idea is so powerful that it can actually result in the development of all kinds of technically like different types of therapeutics. So the question becomes, do we have one entity that kind of becomes all of those things? That's the first option. It becomes kind of a muddy story. 2, do we just like kind of like tamper our curiosity and stick to one thing? Or 3, do we become a sort of hub and spoke model where we have one main company and then we can bring companies out based on certain pieces of technology that we can develop with our core hub? That seems to be what Senti has done here. It's like we have this really powerful hub center, source of discovery, and we can build out spokes.
Right. This is actually speaking to a little bit of a wider challenge that we've seen over the last couple of years too, right? Which is there are all these different companies. They come out with a platform. The platform is amazing. It can do 750 different things. But the problem is you need to actually get one of those assets that your platform generates to the clinic. Mm-hmm. So it's probably cleaner, following that logic, to just spin out a brand new company that is dedicated on bringing one particular asset through to the clinic. That validates the asset, that validates the underlying platform from the parent company, probably. Yeah. And that it just kind of it allows you to kind of separate the two things and yet bolster the reputation of the platform once you've got a success inside of the clinical space.
Yeah. Yeah. And here's more information on this regulator dial platform. It includes a controllable gene therapy for Rett syndrome and controllable armored TILs. What in the actual SBH believes this technology addresses one of the most important challenges. It's a gene therapy? What in the world? Sometimes Senti Biosciences Holdings is SBH. Sometimes companies will use the term gene therapy a little bit loosely.
Yeah.
I would consider CAR T cells to be a cell therapy, right?
Yeah.
But some people consider them to be gene therapies.
Yeah. They're like, it's an ex vivo gene therapy.
Right.
Meh.
Right. It's okay, whatever. It's CAR T cells.
Yeah.
But I get it. If it's autologous, especially, it's a gene therapy. So I'm sure it's a CAR T cell therapy because you can't do a gene therapy for TILs, right? I guess it's in vivo CAR or in vivo, whatever. Anyways, okay. Yeah, super cool. So look out for Senti Biosciences holding new company that's going to be coming out with their Senti 202 asset. And then this is usually a signal for me to keep your eye on Senti because they're going to be pumping out something new really soon. And it's probably going to be super freaking awesome because it's a cool company.
Yeah.
So keep your eye on Senti Biosciences Holdings.
Excellent. Jeff, well, we are a couple minutes early, but we could get our first interview underway if Dr. Sanal is ready.
Sure, let's do it.
Excellent. All right. Well, very excited for this one. Jeff, we have been fans of Coragen for a while. They jumped up the BioTek Voyager rankings a while back, and we've been really excited by what they've been doing ever since. And we reached out to them, and they were kind enough to be like, "Yeah, we'd be interested in coming on the show. Sure, sounds great." So
you guys seem cool.
You guys seem cool. We can cut some clips from it after. So yeah.
Spoiler alert, we are cool.
Well, if we have to say it, then
ask my wife.
Ask my wife and our children. Yeah. Excellent. Okay, well, with no further ado, let's bring her on to the show. Welcome to the show.
Great. Thank you so much for having me.
Thank you so much for coming. We really appreciate your time. If you are, is there a way that you can tilt your camera just a little bit so your head's a little bit more in the center? Perfect. Yeah. There we go. Right on. Excellent. Well, welcome to the BioTek Voyager. We would love to get a little bit of, before we jump into everything that's going on with Coragen, we'd love to get a little bit of an introduction from you and kind of your background and what's kind of brought you to Coragen.
Yeah, well, my pleasure. So first, thank you so much for having me. I have been following the BioTek Voyager for some time. And of course, I saw the July 7th podcast where you discussed about my company, Coragen, right? So I'm so excited to be able to explain more what our technology does and what our company is planning to do, right? So
hopefully we didn't butcher it too bad on the show.
You did actually pretty well. Thank you so much. So a little bit about me. I'm a physician. I was trained in India, and then I also went for my PhD in cancer biology and immunology in the UK. And then I came here as a postdoctoral research fellow at Mass General Hospital, and then moved on to industry. And I work now in cell and gene therapy for over 2 decades. And I've worked in cell therapy exclusively for oncology now for over 11 years. So I'm very excited to be here.
Nice. It's my favorite space of the industry. I love cell therapy and oncology is the coolest spot. So
my favorite space.
Nice. So yeah, what was the very first cell therapy that you got to work on back in the day?
Actually Casgevy. So CRISPR Therapeutics.
Okay, no big deal. Just the first one.
Excellent.
That was such a heady time for cell therapy. CRISPR editing was brand new. We didn't know what to do with it, but we had a great team, and I was so happy even after I left to hear that it was approved. Right, and it's groundbreaking therapy.
For sure.
Fantastic. Okay, so I guess where we always kind of want to start this conversation and go forward from there is basically, getting a good understanding of what is the problem we're dealing with. So when it comes to Coragen, what's the main problem that you're working on? What's the main disease of focus right now?
Absolutely. So what Coragen is trying to tackle, I think, which is not unusual because many companies working in this space are trying to tackle, is very hard to treat solid tumors. So metastatic stage 4 solid tumors which have failed multiple therapies, and the patients don't have any options left. And what Coragen is trying to do, again, we're not unusual. Many other companies are trying to do it, but we have a unique approach. Is solid tumors especially have a very inhibitory tumor microenvironment, and they're deployed as a shield, right? And I was hearing you talk about SentiBio before, right? And so again, many companies are trying to work in how do we go past the shield, and so that effector cells can actually go and kill the solid tumor cells. And so what Coragen is doing is trying to get rid of the shield or minimize the shield so that the patient's own immune system can actually go and kill the cancer cells. So Ben and Jeff, and I heard the podcast from July 7th. I know there was a bit of confusion about whether these are helper cells or they're effector cells, and why we are not actually going after CD8 cells, right? The classical effector cells, the NK cells, the CD8 cells. So
that was me. That was my question.
So as part of this, I prepared for that because all these cells work, by the way, right? And that's one of the reasons why we are not riddled with cancers, because there's this immune surveillance which is ongoing, and it's ongoing throughout life. What cancer cells are very good at doing is they're very good at camouflaging so that the immune cells cannot recognize the cancer cells and kill them, right? Part of this camouflage, and what plays a very important role, are tumor infiltrating Treg cells or regulatory T cells. And if you're familiar with regulatory T cells, they're part of the body's mechanism to prevent autoimmunity, right? So the body needs it. That's why our CD8 positive T cells are not going and attacking our own tissues and organs. Sure. But what cancer cells do, and especially solid tumors are very good at, is they co-opt this system and use it as part of a shield, right? And they do not allow the immune system, whether it's the endogenous immune system or its CAR T cell, to recognize and attack and kill the cancer cells. And that's what Coragen is targeting. Coragen is targeting these tumor-infiltrating Treg cells.
So does it have to do with the enhanced ability for Tregs to traffic into tumors versus like the CTLs?
So just like any other type of immune cell, Treg cells traffic to tumors, right? So they will also pass through lymph nodes, right? So they will traffic around the body, and then they'll also be resident in the tumor itself. So they're not doing anything unusual.
Right. So then why? So again, I'll ask, why not delete Circ3 from like a CAR T cell, like a CD8 CAR T cell? Why do it with this kind of roundabout way of doing Tregs?
Because, and let me go a step back, right? So our technology has come out of the Baylor School of Medicine. The late Dr. Bert O'Malley is considered the father of molecular endocrinology, and his lab discovered this family of proteins called steroid receptor coactivators. And one of them, the one we're targeting, which is SRC3, is overexpressed in regulatory T cells more than other cells, right?
Okay.
And what Dr. Bert O'Malley's lab found was that not only is it overexpressed, it's also a master regulator gene, which then has a downstream effect on multiple immune checkpoint proteins, right? So it's, instead of targeting individual immune checkpoint proteins, which is what immune checkpoint inhibitors do, if you delete or disrupt this master gene regulator, you're actually downregulating many of these other immune checkpoint proteins, which are known to be overexpressed in solid tumors on Treg cells. So it's a very elegant biology, right? You're basically getting to the root of the problem.
Right. And I think the list was like, sorry, what was that?
Yeah, then that's what Coragen, that's what our investigational drug is based on, this knockout or disruption of this master gene regulator, which is upstream of like dozens of immune checkpoint proteins.
Yeah, it's like PD1, CTLA4, like all the ones that everyone's got,
right.
Like three, all the we have monoclonal antibodies against half these things. And if SRC3 is the one who's responsible, at least in Tregs, for causing the expression of those, you can essentially administer like 7 or 8 different therapeutics, quote unquote, by just inhibiting Circ3.
You're absolutely right, and that's our goal.
Nice.
So then I guess a follow-up question would be: Is the SRC3 mediated expression of PD1 of CTLA4 just not as prominent or prevalent a mechanism within other kinds of immune cells? Is it mainly in Tregs?
It's definitely overexpressed in Tregs as compared to, example, NK cells and CD8 positive T cells. And this is especially prominent in the tumor microenvironment, which is why this was such an attractive target for us.
Nice. So yeah, if we back up here just a little bit, right, when we're kind of looking at your approach for solid tumors, obviously solid tumors haven't had a lot of success when it comes to the other therapeutic approaches that have been tried. I'd be curious to kind of get your thoughts, right? Like, why do you think it is that so far we've had such a challenge with CAR T therapies not working super well, or some of these different approaches that have been taken? And then separate from that, or following on from that, what kind of the unique mechanism would be here with Tregs?
Absolutely. So, and I've actually worked in CAR T cells and solid tumors in the past, right, with mixed success. And what stopped those CAR T cells from working in most patients, but were successful in a few patients, was the tumor microenvironment, right? So if you look at effector cells, which CAR T cells are supercharged effector cells, they can kill cancer cells, but they're prevented from doing so, right? So, and so it's almost like it's synergistic. So you have either the effector cells, the endogenous effector cells, or you have the supercharged CAR T cells. They can all do what they're supposed to do, but they're prevented from doing so, right? So what Origin is changing, or I should call it CRG-150, which is our first investigational product, is this what prevents these immune cells from killing the cancer cells?
So okay. The SRC3 knockout Tregs go in, and they can help to repolarize the microenvironment, and then that is going to then be the actual therapeutic effect would be dependent upon other immune cells actually being the effector cells.
Absolutely right.
Does your therapy require lymphodepletion?
No, it does not. So when you ask me what is differentiating about our approaches, we do not require preconditioning by lymphodepletion. And that is very important because we actually need an intact immune system, a functional intact immune system, to be able to kill the cancer cells. So actually lymphodepletion would do the exact opposite, right? So that's one very important differentiator, and that ties into the patient experience. I mean, these are stage 4 cancer patients. They are going through so much already. And then to go through lymphodepletion, then you have all the side effects which are related to these supercharged effector cells. Example, cytokine release syndrome, neurotoxicity, and many others, right? We have not seen any proof, at least in our non-clinical studies and multiple non-clinical studies, that we are going to see similar side effect profile. So no preconditioning by lymphodepletion, hopefully safer, right? As we're hoping to prove in our upcoming clinical trials, and effective. Yeah. Because it's basically leveraging a fundamental mechanism of solid tumors.
So I don't want to get too deep into like the nerdy science, even though I totally want to get too
proceeds to immediately go deep into the nerdy science.
Just like dig as deep as you can. Do you have any data that you can discuss that implicates the innate arm versus the adaptive arm upon like infusion of your cells? I would go as far to guess I would expect to see a lot of innate immunity, macrophage repolarization, dendritic cell activation, and things like that.
Yes, you're right. And we've shown this in multiple preclinical studies. And actually, Dr. Bert O'Malley's lab has published dozens of papers. I mean, dozens and dozens. And so, and if you read those papers, and I'm happy to send them afterwards, right? You do see this repolarization, exactly as you said, the macrophages, the NK cells, but also an infiltration of CD4 and CD8 cells.
Yeah.
So it's actually multi-pronged.
Yeah, the CD4s will just get in there and just start to like create a feast. Right. Yes. They're really good at just like making a bunch of different cytokines. And if you can polarize them the right way, then it can be like really, really good. Just that in cancer, it's like the different metabolites and whatever in the tumor microenvironment can make them like really harsh Tregs.
Yes.
Yeah. Excellent. Well, folks, for everyone else who is listening, if you have any questions for Sonal, I'm sure she will take some. If you have any questions, assuming that you're not asking for proprietary details, I'm sure she would take some questions. So if you have any questions, feel free to put them in the chat. But I guess if we can just back up for 1 second, Sonal, with regards to kind of the standard approach of CAR T therapies versus Tregs, and I love how you've been able to talk about this so far, right? You really do have the difference between like a grunt and a diplomat. You have the difference between like the frontline soldier versus the general who's kind of like orchestrating the larger moves. I'm curious if there are any other analogies that you guys kind of use to kind of compare and contrast these two different approaches, right? Because Tregs just aren't as well known as CAR T cells yet.
Exactly, right. And so I call it the stealth mode, right? They're there, they're everywhere, they're in every solid tumor, right? And as you can see, when you think about immune checkpoint blockade and why it's been so successful, largely because it does act on these overexpressed immune checkpoint proteins in regulatory T cells, right? So there's already proof, both investigational and real world proof that this works, right? So I call it the stealth mode. And I find that very, very interesting and exciting that it is already existing. It's everywhere. It's prevalent, and we are able to target that.
There's even been a lot of evidence that shows that even with a CAR T cell population, you need to have a good population of CD4s in there as well. Otherwise, you don't get as much activity from your therapeutic. So they're obviously doing something in there, and it isn't as the story of like a cytotoxic T lymphocyte CAR expressing CTL is just easier to understand, right? Yeah. Like we send this thing in and it's like a gun just to shoot a cancer cell and it dies, right? But with the CD4 aspect, it gets a little more complicated and people are a little bit less averse to, knowing what it is. But they really are like, they're there for a reason. Evolution has given us the cell population for a reason. And it seems like they are very powerful therapeutically.
Agreed.
So
tumor immunology is fascinating, right?
Yeah. Yeah. I guess one other component of this too then. So like when we start to get into the data of this approach, right? Like what were some of these early pieces of data that you guys saw that just really made you go, "Oh shoot, this is good."
Right. Absolutely. So obviously the overexpression of SRC3 in Tregs, that was an early discovery. And because there was already earlier data to show that knocking out SRC3, example using a small molecule inhibitor, was actually a very powerful tool in eradicating cancer. So what the Baylor lab did was they put those two pieces together and came up with this gene-modified autologous ex vivo edited Treg cell therapy, and that's how we are where we are right now.
So could there be potential to do things like in vivo, gene silencing? Would that be maybe less manufacturing, burden?
Yes, and if you look at our updated webpage actually and look at our pipeline, we do have an in vivo approach that we're exploring. I'm not allowed to talk much about it because it's still, as you said, proprietary, and also an allogeneic approach. And there is, we have so much preclinical data, and not just in the three tumors we're actually targeting. We're targeting HR positive, HER2 negative breast cancer, which is the most common breast cancer subtype, but also the highly aggressive triple negative breast cancer and metastatic prostate cancer, right? But we also have preclinical data on, everyone knows about pancreatic cancer, glioblastoma, right? They are like they're death sentences basically, right?
Yeah.
We have pretty compelling preclinical data on those cancers also, as well as colorectal carcinoma, melanoma, and non-small cell lung carcinoma. And all of that is available on our webpage, right? So there's this universal approach because we're not targeting anything on the cancer cells, right? There's no target, there's no PD-1, there's no PDL1 or CTLA4. We're going after something which is fundamental in solid tumors.
Yeah, I'd be very curious to know your glioblastoma data because I did some GBM stuff in my postdoc, and I was shocked to learn that sometimes in some cases, the majority of the tumor mass is actually macrophages. And they're very polarized to being immunosuppressive. If you can and there's a lot of established mechanisms of doing like intratumoral or intracranial cell therapy infusions. Getting a big bolus of immunoactivating Treg cells, or I can't help but call them CD4 helpers, into that tumor. Imagine if you could take even 10% of those macrophages and repolarize them.
It's going to be very powerful,
it's game over for the tumor.
Exactly right, and this is as you know a challenge with many what we call cold solid tumors, right? And if you can repolarize them so that now they become what I call hot tumors because of this infiltration, increase infiltration, right? That is what
GBM is like ice cold, like a popsicle. There ain't nothing going on out there.
Agree.
Okay, cool. So I guess the other component of this that I'd like to dig a little bit deeper into as well, right? Just to, because ultimately we would love to make some clips from you as well with regards to this. How exactly, what is the mechanism here by which you don't require lymphodepletion the way that so many of these other therapies do, right? This is something that I've always been somewhat troubled by, right? Is you've got these incredible cell therapies, we're utilizing our own immune system, and yet there is this big flaw in so many of them in that you require this lymphodepletion step. So why is it that lymphodepletion isn't required with the Treg therapy?
Because of a different mechanism of action. So ours is an indirect mechanism of action where we require a functional endogenous immune system, and what I call a polyclonal approach, right? We're not requiring something which is just one clone of cells which is expanding. It's the body has a repertoire of an immune system which has already been exposed to the tumor, and as I said, which is perfectly capable of killing the tumor, but it's prevented, right? It's almost, I call it like the Treg cells act like a bouncer at a nightclub, and I've also heard it being described like that, and they do not allow these immune cells to enter. And what we do is we're getting rid of that velvet rope, and allowing those immune cells. So you do not want to lymphodeplete because then those endogenous cells are gone. Our Treg cells by themselves do not kill the tumors, right? They make it possible, that they permit.
Don't tell the investors that, though. They might not understand.
they permit the body's own immune system to actually come in and kill the tumors.
And it isn't.
Right.
Yes, and it isn't that CAR T cells necessarily need lymphodepletion. It's that they function better with it.
Exactly.
Right? Whereas because of the difference in mechanism of action here, it actually would be the opposite. Whereas our therapy actually functions better without it. And being autologous, there's really no and like with an autologous cell therapy like this, where you're deleting something, you're not going to express some kind of immunogenic thing on your cells. There's no risk of rejection, really. The allogeneic, just give them some stealth technology and you're good to go.
Yes, absolutely. We do not have any risk of graft versus host disease, right? Which is, again, something which has been reported with allogeneic CAR T cells and, has held them back, right?
Yeah. It's super, super cool. And again, it's one of those things where you're, the standard narrative is this cytotoxic T cell, linear mechanism of action. The immune system isn't linear. It's a matrix, and to limit it to hey, we have this one linear thing that we're gonna, here's our MOA. It really doesn't take advantage of the intrinsic ability of our immune system to kill cancer, which you mentioned. Most people, I've heard studies where they've taken autopsies of car accident victims, and they find that a lot of people actually have tumors. It's just that they're in the immune surveillance state.
Exactly.
Right, and our immune system can take care of these things.
It's very powerful.
Yeah, and it's just giving it a little bit of a spark can go a long way.
Yes, absolutely. And please understand, right? It's not us versus them. So it's not like CRG 150 versus CAR T cells. I think what we want to offer is a toolbox for patients, right?
Yeah.
And for oncologists, so that they have choices to treat patients using something which is effective, but also safe, right? Cancer patients deserve to have safe therapies also. Right? Not just effective.
Agreed.
Excellent. Cool. Well, folks, if you have any questions for Sanal, we've still got her for around 10 minutes here, so feel free to make your questions known now. But I guess just with regards to the specific signal that has surfaced you on the BioTek Voyager as well, right? You've started this IND clearance for a phase 1/2 trial. So when did this first get initiated? When did you guys first file for an IND?
We filed for the IND in May, late
Excellent.
May, and
Fantastic.
we got our clearance exactly 30 days later.
Perfect. On the money. And then I guess where are you planning on doing recruitment for this? Is this based in just one state? Or are you planning on doing multiple clinical sites? What does that look like? Or if you're allowed to say.
Yeah. So we are planning multiple clinical sites throughout the US. Sites which are not just see a lot of solid tumor patients, but also have a lot of cell therapy experience. So, at this point, I'm not allowed to say what the sites are, but they are major academic centers. And our goal is to provide geographic distribution so patients don't have to travel too far.
So are you in Texas?
Well, our company's headquartered in Texas, yes.
Nice. Okay.
I'm personally based outside Boston.
Of course you are. Isn't everybody?
Except for Ben and Jeff.
Yeah. I'm in the mountains. Ben's in the Arctic.
Well, that's one way to talk about Canada. Sure. The Arctic. Sidebar. It's actually we've had like massive forest fires up here. So like yesterday, it was like a yellowish hue everywhere, and you couldn't see the sun.
It's like we're in hell.
We actually have the smoke plume over Boston also.
Oh yeah. Wow. Sorry about that. I feel like that's the standard Canadian answer at this point. Oh, cool. All right. Excellent. Very good. So I guess like in terms of next steps here, I know you've talked a little bit about like your allogeneic approach. You're going to be looking into potentially like an in vivo approach as well, and you can't say too much there. But yeah, just in terms of like what's next for the company, aside from this clinical trial, is there anything else that's in the works that you can speak to without, everyone signing an NDA?
So I think one of the most important things we want to do is do it right? So we have this one clinical trial that's going to be a basket study with all 3 indications at the same time. We want to see what our recommended phase 2 dose is by going through dose escalation, the phase 1's part of the study, selecting that recommended phase 2 dose, and then going into cohort expansion. That really is our primary goal. And so my goal is not to have multiple studies, it's to have this one study and do it right. When we have a safe, efficacious signal, our goal is to actually expand into additional indications. So that's where the pancreatic cancer and the glioblastoma come in. Right?
Nice. It's very odd, but very promising to see a basket trial in oncology for a T-cell therapy. So congrats on that. That's
Thank you. I'm very thankful. We pushed forward and the FDA went and agreed.
Yeah. Excellent. And again, the mechanism of action is universal. So it makes sense. I mean, just do it in everything, right?
Exactly.
But yeah, I mean, like you're hitting some big ones. Triple negative breast cancer, prostate cancer, and what else?
HR positive.
HR positive. Yeah. HER2 negative. Why not HER2 positive? Just out of curiosity.
At this point, I think there's so many effective therapies for HER2 positive.
Okay, sure.
Yes, and that's one of the reasons why we decided to go with the indications we have. It's not closing the door, but it's not going in right now.
Sure.
Perfect. Okay. I'm a manufacturing guy at heart. That's kind of where I got my start. A little bit more on the gene therapy side, but I've spent some time on the cell therapy side as well. So yeah, like what does manufacturing look like for Tregs, right? Most people would probably kind of think of kind of your standard 14 day process for a CAR T therapy. What does a Treg therapy look like for taking it from the patient and then manufacturing it?
I have to give a big shout out to our Chief Technology Officer, P. Jalinas, who has basically taken a rare cell, because Tregs are rare cells, right? And they are not exactly easy to manufacture or expand. But we have this bespoke manufacturing process, which allows us to expand these edited Tregs to actually make those and make more, if I can say that, right? And as was in our press release, we have partnered with Lonza. Lonza has decades of experience in this field. They are, if I could say, the leaders in cell and gene therapy manufacturing. So it's this internal technology plus having partnership with Lonza. And so we are able to consistently manufacture sufficient doses to actually to be able to dose even at the highest dose level that we have planned.
Excellent. And I don't know if this is something you can share, if this is proprietary, so you can just tell me to shut up. But like, how long does it take compared to a CAR T process? Is it longer, shorter? I imagine, right, like you've got fewer cells to work with, so that makes it a bit more challenging.
Very standard. The time is actually very standard. Right now, we're targeting 28-day vein-to-vein time, which is the industry standard, right?
Yeah. That's awesome.
Yes.
Phenomenal. Perfect. Excellent. Well, we've just got a couple minutes left here, folks. If you have any final questions, you can get them in. But I guess there is one question we try to ask each of our guests. And your answer can be a conglomeration of everything we've talked about already, or it can be something else entirely. But the question that we ask is, what's next in biotech? And the idea being here basically like, what's slept on in biotech that's going to be a big deal in a couple of years?
So it's what you mentioned earlier, right? It's people are targeting the soldiers, right? But not necessarily thinking about the generals. So Treg engineering, I would say, is something which is an unmet need, if I could say that, right? I mean, immune checkpoint inhibitors do target them, but more like single immune checkpoint immune checkpoint proteins, right? So I believe that Treg engineering is going to be the next big step. And then manufacturing and access, right? Having a shorter manufacturing process and making it accessible to patients, not having lymphodepleting chemotherapy, for instance, having manageable safety profile, right? That all of that will actually make this not something which is niche and only available at major academic centers, right? But actually something which is more global. I think that is that's the next wave.
Yeah, and then we had Carl Schulhammer from Desi Bio on, I think 2 weeks ago, and he said that one of the biggest things that he's looking forward to is physician awareness, because one of the problem with like cell therapy administration or use, if you want to use that word, isn't manufacturing, it isn't safety, it isn't X, Y, and Z, it's prescriptions. It's physician awareness and prescriptions. I'm wondering if like the lack of lymphodepletion and the less immunologically toxic profile of Tregs would maybe put down some barriers on that front, to be like, hey, this is not going to be like something super harsh for your patients and they won't hate you for putting them through it.
That's what has actually got a lot of our investigators excited. So when we started reaching out to these major academic centers, that was the calling card, right? But there's no lymphodepletion, and then the fact that this is potentially safer, but hopefully as effective as we've seen in our preclinical studies. In preclinical studies, we've seen eradication of the tumor, and when we re-challenge these mice with the tumor again, they do not develop do not develop cancers again, right?
Yeah.
And so it's a combination of all of that which has made this very attractive.
Well, looking forward to the progress for sure.
Yes. Yeah. I first remember learning about Tregs when I entered cell therapy, and I was like, man, these are just so much more elegant in some ways. Like you need both for sure. But like they're just such an elegant approach. So we really appreciate you coming on and sharing more. We're very excited to see where you guys go with this. And yeah, we're very excited by the progress you guys have been able to make so far, and we will be following along with great interest.
Great. Thank you so much for having me. This was a lot of fun.
Looking forward to that first patient dosed press release. That'll be the next co-regen signal.
Try my best.
Yeah. Excellent. Sounds good. Well, thank you again, Sonal, for coming on, and we'll see you around. Thank you so much.
Thank you so much.
Cheers. All right. All right. Excellent. Well, Jeff, I love that. Perfect. Again, I love Treg so much. It's awesome to see what they're doing.
If I had to like make a leaderboard of Ben's favorite biotech analogies, it would be number one would be T cells, CD8s are soldiers, and Tregs are diplomats. It's like whenever you get the opportunity to use it, it comes out immediately.
I made a LinkedIn post once about that, and I've like reused it like 3 or 4 times. Yeah. So it's just kind of like the top of mind example every single time. So that is part of the reason why. Excellent. Well, Jeff, today is a great day because we have not one cell therapy developer, but 2 on the show, and we're going to be talking to Ramo next in a very similar application. Just very quickly here, this one is on the neurology side as it relates to like brain injuries. So kind of on the opposite side, we've talked about cancer so far. Now we're going to be talking about something that's a bit more rare, but of course, just as important. So yeah, very excited for that conversation. Should we bring Ramo on, Jeff?
Yes, sir. Do it.
Excellent. All right. Well, with no further ado, proceeds to give him a do. Oh no! There we go. There we go. We're good at this. All Ramo, right. that was an awful handoff. You did great. Thank you for that.
That's fine. No worries.
We are professionals. we know what we're doing. But yes, thank you for coming on. How you been?
Very good. Thanks for having me on. Congratulations on BioTek Voyager. It's great. I appreciate what you're doing, and I'm all for Ben renaming everything, like new nomenclature according to Ben for every cell therapy. I think that's a movement I can get behind.
and watch it catch on. I'll be so annoyed if Ben has that kind of pull in the industry. It's like,
I don't.
Why does everyone care what Ben thinks? I exist too.
I once made a post about maybe we should call CAR T therapies something else, just because CAR is kind of weird, and Brian Levine was like, no.
Yeah, that one didn't go over very well.
No.
I remember that. Everyone's like, shut your mouth, kid.
Yeah, fair enough. Anyway, excellent. Remo, great to see you again. It's been a while since we caught up. Would love to kind of learn about what's been going on in your life and what the big problem is that we're tackling here.
Yeah, great. Thanks for having me. Just a little bit of context and history. As Ben and Jeff, you guys know me for a few years now in the context of Cytonus Therapeutics. So several weeks ago, right before Bio International, we announced a formal name change to Axiom Biosciences. And we've still been around for 8 years, chugging along. The reason why that was important for us was because we were coming out with some clinical trial results that were not based on the asset class that we were known for at Cytonus, just because the asset class that we had at the time at Cytonus had a lot of interest, and during our development stages, we didn't think it was important to take the eye off the ball for people. They were engaged in that, but in the background, we were developing multiple different types of delivery platforms. So Axiom Biosciences is still fundamentally based on delivery and building platforms for delivery. So we had done a strategic deal in 2023 with a Korean partner called Medino, and we co-developed a mesenchymal stem cell product for regenerating damaged tissues, in particular in brain injuries. And at the time, we had just started our phase 1 clinical study, and of course, it took a while for that study to finalize and complete. And it was in 2 rare pediatric diseases that we have 2 rare pediatric disease designations for now. And right around the end of last year, we closed out the study. The results were fantastic. And in the next several months, we were working on a number of corporate strategies as regulatory strategies, how to move forward. But we were really proud of the announcement that was released earlier this week about our phase 1 studies and how we're moving the asset forward.
Fantastic. So yeah, basically, so I guess if I remember correctly, right, previously you're primarily focusing on mRNA LNP delivery. Is the basis for this still the same, or is it slightly different?
Yeah, no, this is not that asset class. This is a stem cell. It's a mesenchymal stem cell taken from the Wharton jelly origin of umbilical cords in healthy newborns. And what makes the product different and differentiated is the fact that in our manufacturing process, we essentially condition these cells prior to giving them to a patient in order for us to initiate a natural process of having cells start to go down, especially stem cells, go down a specific pathway. So I think that this was something very much missing in the history of stem cells. And it is one lever of control that we felt was very important in the context of trying to develop platforms for then multiple different indications.
So how does this thing work? You have these MSCs, and you do direct CNS infusions? And then what
right.
happens?
So, yeah. So, Jeff, let me back up. Let me tell you about the clinical trial, because the second part of that makes a little bit more sense. So, there are two rare pediatric diseases. One is called intraventricular hemorrhage of newborns, and the other one is called hypoxic ischemic encephalopathy, again, of newborns. It's most people don't know these two conditions, but they are rare diseases, although they're not uncommon. They do not have any sort of genetic component, and it's super unfortunate because these are devastating, lifelong problems for not only these children, but their families if they survive moving forward. So IVH is essentially equivalent to having a hemorrhagic stroke. The only association with IVH is prematurity. So if you're born less than certain week of gestation and you have a really low birth rate, the probability of having mild or severe IVH is very high. Hypoxic ischemic encephalopathy, on the flip side, is a otherwise full gestational birth through natural birth process. And this is a very classic example of the umbilical cord getting strangulated around the child's neck. Essentially, it's an asphyxiation. And ultimately, that insult is an ischemic insult.
Sure.
Nevertheless, both the brains are permanently damaged. In IVH, it's very unfortunate because there's a very high level of mortality associated with these children. So in Korea, where we did the phase 1 clinical study, the natural history of these children should be around 46% of them will not see the second week of life. And in our clinical study result, we had no mortality after 12 months, and followed the children for developmental measurements up to 24 months. And on the flip side, with HIE, these children typically do not have mortality associated, but they have severe morbidity. So a lot of these children will have cerebral palsy. Across both classes of these newborns, they will have very high percentage, like 70% of these kids will need long-term care. They have developmental delays. They have physical problems as well. Some of them will manifest in blindness, in deafness, and so forth. So the studies are a little bit complicated because you have to let these children grow in order for you to figure out how they're comparing to their, their otherwise peers that don't have this problem.
So could you use this post-stroke in adults?
Yeah, we're going there, Jeff. So exactly the point. Although we tested these in these two rare pediatric diseases, the next step is to naturally take them into stroke. So kind of going back to the outcomes, the clinical trial outcomes, and why we think this is not just feasible, but really exciting for us from a perspective of scale, is that the two insults, the primary insults, are the same, right? It's either ischemia or it's a hemorrhage, and ultimately the brain tissue is permanently damaged. And what we did, and I don't think I answered this first time, Jeff, I'm sorry, but what we do with the product and how it works is that we condition the cells prior to administration, and we do direct intraventricular placement of these cells. It's very easy. It's relatively easy in children because the bony structure of the skull is not yet fused. So there is a pathway. In adults, really, we're talking about severe strokes and severe hemorrhagic strokes as well. So ultimately, those individuals will likely be in ICUs with ventriculostomy tubes. So the pathway is likely there, but it's not a very complicated thing to do from an administrative perspective.
Mm-hmm.
And ultimately, the product, the reason why we condition them is that we are putting them in a simulated environment where they are expected to see ischemic tissues in the brain.
Mm-hmm.
So if we back up, what's the name of the first injury that you're going after? The one that has the really high mortality rate?
Intraventricular hemorrhage of newborns, or IVH.
Okay. Gotcha.
Yes.
IVH. So with this one in particular, so I just want to make sure I'm understanding this correctly. Normally, roughly half of these children don't make it past the second week of life. And with the group that you guys have been able to treat from this, you didn't have any who passed away, or how does that work? I just want to make sure I've got the data correct there.
Right, exactly. So depending on where you are, and I think the mortality has a lot to do with the downstream infrastructure of healthcare systems, NICUs in particular. But the numbers range from the low of like 35% to 60% internationally. In Korea, in particular, where we did the study, that number lands at 46%. So we dosed 5 children in that arm, and we got no mortality at all. And these are severe IVH. So these are not.
Ramo, let's go.
Yeah.
That's awesome.
So these, and so that mortality perspective is just a really good initial indication of the product working. But as we look forward, we were even more impressed with some of the other aspects of functional improvement. So none of these children needed any anti-epileptic drugs through their development. This is a huge thing. Wow. We got no cerebral palsy in these kids as well. And the neurodevelopmental delays were dramatically, statistically significantly reduced. So I think ultimately what gets us all really excited is that we're talking about a quality of life that is now instilled in a newborn for potentially, hopefully 80 plus years.
Right. Potentially life.
Given 1.5 weeks of life. That's incredible. Wonderful. Amazing. And so I guess when did this trial take place?
It's been ongoing for the last 3 years.
Is this an allogeneic thing, or is it autologous? So they use the baby's own umbilical cord as the source, or is it just like banked?
No, it's allogeneic. So we have a really good supply agreement for umbilical cord MSCs, and we could use those as master cell banks.
Yeah. So is it true that umbilical cord blood is considered medical waste?
Is it true? Historically, yes. But there is an industry where people bank these cells.
Yeah,
so
for sure. Well, I don't mean it in like a. I mean, like, it's probably easy to get a hold of.
Yeah, it's not complicated, right? There are very established companies that actually will either offer you to bank your cells for the entirety of your life, or if you don't want them, they'll just take them as a donation. And it's actually really good because they put them into the circulation for people who need stem cell transplants. So this becomes like an international collaboration with people who need these types of transplants all over the world. So they are very readable.
So when the MSCs get in there, are they actually like developing into neurons, or what do they do? Does it matter? Do you know?
Yeah, so there's no real indication that they differentiate. That's important. Okay. I think one thing to understand is that, and we've been working with stem cells for 8 years, that every stem cell is different, meaning the source really differentiates the characteristics of the stem cell, although they may be characterized as MSCs. An MSC from a bone marrow donor is very different in characteristics from an adipose-derived MSC is very different than an umbilical cord MSC, and also iPSC-derived MSCs as well. The umbilical cord MSCs, the reason why we love them is because universally they're accepted as being probably the best for regenerative medicine because they are arguably the healthiest, youngest cells in the human body. They literally are 100% derived from the child and they're harvested at birth.
Okay. Super cool. And so in terms of like the delivery of the like is there a genetic payload that is getting put
how
in these?
How do they not get rejected?
Right. Well, so MSCs are immune privileged. They have very little MHC1s and have no MHC2 proteins. So they've been in like 1,500 clinical trials. So they have this very good compatibility for being given to people. But the other part of it is, what do they do? So the mechanism of action is through their paracrine functions. That they upregulate HGF and TIMP2, which are both growth factors that protect dying cells, but they're also anti-inflammatory. So they're halting this, and ultimately it's regenerating otherwise brain. One thing I learned about brain is it's, as we all know, it's super sensitive. I didn't quite know how sensitive it is. There's a number that shocked me. For every minute of ischemia, 1.9 million brain cells die.
Whoa.
Yeah. So when you consider us in like an adult stroke, which more people are familiar with, you know, there's a reason why there's a stroke code, because the time is of unbelievable value to the patient. But never underestimate, you know, any signs and symptoms of stroke. You know, I was a physician in my first career, and, you know, I'd always be surprised how many people just tried to sleep through a problem that they shouldn't have, right? And it's devastating.
Man. Okay. So if we back up here for just a second, then you've got someone who you've got either a baby who is suffering from this, or you have an adult patient. You're looking at 1.9 million cells dying per minute. How quickly can this therapy be administered to reverse that?
Great question. In children, it's widely believed the sooner the better. Paradoxically, in adults, there is a period of time where you can act for regenerative therapies. So there is a inflammatory cascade in adults that you essentially need to kind of start before you can give a stem cell. And people have demonstrated this in the past, that it seemingly doesn't work if you give it too quickly. In children, we don't take that risk. So I think there's a neuroplasticity issue with children. So we want to intervene as quick as possible. Our product can be administered anywhere from 3 to 7 days. It can be administered ideally up to the first 14 days after diagnosis. And the diagnosis is usually within the first few days of birth, if not at birth.
Right. Right.
Gotcha. I guess that was my question. Like, do you like I don't know, like as mom is giving birth, is this something that's diagnosed, or like, oh, your child is experiencing a stroke, and so as soon as you're delivered, we're going to administer this therapy, or instead this is something that kind of happens post delivery. We realize this has happened. Oh shoot, let's get this therapy into this kid.
Yeah, with IVH, it's clearly triggered by prematurity. So if you're a premature birth, the staff is already keen on figuring out all of the problems with the child. So they routinely diagnose this with ultrasound. Okay. And that's one thing. The pathology of IVH is essentially that these blood vessels are so premature that they're very frail. And one of the things that, you do for these kids, because there are no treatments for either of these, but you simply don't manipulate the child. So very minimal movement of the child is important in those first few days, right? Because any sort of trauma will increase either a existing bleed or cause a bleed. So, staff NICUs, are really well aware once it's a premature birth, there's protocols that are implemented. So I don't think the diagnosis is really the problem there. It's a question of, how quickly people want to intervene. And when I say people, I wholeheartedly believe the healthcare system wants to intervene. But there's a decision the parents have to make.
Right. Yeah. Oh, well, this is a timely conversation because my wife is going to give birth in a couple weeks. And
I'm sorry, Ben, and I knew that, and I was like, I hope you're not reading too much into this.
No, I prefer to go in with my eyes open and just kind of be aware of things. So this is good to know. Yeah. Okay. So this is great. So talk to me a little bit more about like this process now. If you're going to be able to take the same application and bring it into strokes for adults, this isn't something that's required necessarily right away. Like what's kind of driving that? Why is this something that wouldn't work as well, immediately post stroke?
Yeah. So first, we're probably more realistically looking at severe strokes, and there just is an inflammatory process that has to set in order for stem cells to be effective. And other people have tried this in the past, albeit with a different product, but nevertheless, still a stem cell. And that's kind of a window of opportunity for commercial scale, because in a rare pediatric disease, it requires downstream thinking about how do you scale this commercially. But in ischemic stroke, which affects 780,000 Americans annually here in the States, it's about 12 million globally. So it's a huge, huge problem. And right now, the only therapy that is available to them is TPA. So it's an anti-clogging therapy. So they want to reperfuse the brain, which interestingly enough also cause a little bit of reperfusion injury. So there is always going to be damage that potentially needs some regenerative capability.
Okay, interesting. Yeah, it shows you I don't know a whole lot about strokes. It's really cool, just kind of like the breadth of different things that we get to talk about on this show. This is it's super interesting. Okay, so this is all really cool. You've been able to do these clinical trials so far inside of China, or sorry, not inside of China, inside of Korea, inside of Korea. So, like, what are the next steps for the company? What should we be looking for next? What are the next steps from a clinical perspective, I guess I should say?
Yeah, we're looking to advance the asset into the next stages of clinical development. So that's in process right now. We're also looking to expand the indication. So we have one product, and now we're expanding it into 4 different indications that we have data on, that being IVH, HIE, ischemic stroke, and hemorrhagic stroke. But potentially, the process and our specialty cocktail of developing the preconditioned media lends itself to other sorts of ischemic damage, potentially in the heart. There's brain damage that comes from people who have cardiac arrest as well. So that 1 minute number is actually really impactful when you think about all of the problems when it comes to circulation.
Okay. And what is the dose size here? If you're losing, what is it, 1.9 million cells per minute inside of one of these ischemic events, what's the size of the dose that you're typically delivering?
Well, I don't quite remember off the top of my head. Sorry, but it's I can get that back to you. It's manufacturally very manageable. Yeah.
Gotcha.
So for every donor, we have several hundreds of products that can come from. So we don't really have a manufacturing issue, although we are a cell therapy. But there is also something to be said about the simplicity of this, right?
Right.
And I know we are all super enamored by all of our tools and how we can use them at times, but sometimes it's worth just stepping back and looking at, why some of these problems didn't work in the hands of others. And you can very quickly come up with several relatively easy things to address before you start engineering, really complicated cell therapies.
So I missed the discussion. Is there any kind of genetic engineering or no?
No,
Sorry.
There's no genetic engineering.
Okay, right. So you just take them from the bag and just put them in. Do you expand them at all?
We expand them. In the process, we condition them to have certain factors that simulate the environment we're going to be introducing them into. And I think that's important. It sounds like a small thing, but I actually would argue it's a very big thing in the last 4 decades of stem cells being tried for many reasons. If you look at the approach in which others have done this, they essentially take naive stem cells and they give them systemically. And ultimately, you're relying on the fact that stem cell is going to go to the condition you want it to go to and then produce a factor you want it to produce. But as everyone knows, all 3 of us on this call, right this moment sitting here, we probably all have at least 10 inflammatory things going on in our body, and stem cells naturally have tropisms to those, right? So there's a level of control that most groups have never really addressed that I think is very important in differentiating our approach. So not only do we control the product, but choosing to go to the brain is another level of control because it is evolutionarily protected and sequestered from the rest of the body, not only from the blood-brain barrier, but from a bony structure. Sure. So for us, it was a very deliberate and well thought out approach towards how do we address the things we can address that are relatively simple to kind of get to. And then if the asset works, if the product works, great.
Love it.
That would be great.
Yeah.
That's awesome. Perfect. All right. Well, I want to be we want to be super respectful of your time, Remo. There is one question we try to ask each guest at the end, and you may have already heard it from the previous guest on the show. What's slept on in biotech right now that's going to be a big deal in a couple of years?
Yeah, it's a great question. I'm gonna answer this from, let's say, corporate way, right? Not like a technology, and I know this might be boring to you, but for the last 3 or 4 years, we've seen this movement of capital and interest towards assets, right? And away from platforms. And I've always thought, and we've never strayed from this, that this was short-sighted. I believe in the future, pendulum's going to go back and people are going to realize, well, if one asset works, then if there's a platform behind it, it's going to be really, really important. So I think looking forward, I believe that, the last 3 years are not an indication of the year or two ahead of us.
I'll take it. Right on. Excellent. Well, we'll take a macro trend, and that's a good thing. Fantastic. So yeah, keep that in mind, folks. Platforms are out. Assets are in.
No, platforms are in. Assets are out.
Yeah. Oh, sorry. Yes. Sorry. I'm clearly tired from my 2 kids. Excellent. Gotcha. Well, fantastic. Thank you so much, Remo, for coming on. We really appreciate your time, and we will be looking forward to the to following announcements, I understand, in the weeks to come that you can't be spoken about now, but some interesting stuff in the works.
Yes. Thank you, guys. I appreciate your time. Congratulations on BioTek Voyager again.
Oh, well, thanks. Thank you very much. We appreciate you coming. Thanks, Remo. Cheers.
I know.
Excellent. All right. So, Jeff, that's this. I was really impressed to figure out that there's not any genetic manipulations. You're basically just growing the cells, conditioning them slightly, and then you're off to the races.
Well, a little bit off topic from that. Whenever we talk about anything new, I'm always like, "Oh my gosh, do I have this?" And the entire time he was talking, I was like, "Have I ever had a stroke? Like, could I have had a stroke? I haven't thought of strokes in forever." And it's like, it's like medical school syndrome. You learn about a new thing, and it's like, I probably have had that like yesterday.
Well, I remember a couple of years too. Maybe it was you. No, I think it was before I'd met you. I learned this far too late in my career. I should have known this earlier. Like in the extent of the conversation that we've had over the last couple minutes, like we have both gotten cancer and then it's gone again.
Right. And I had a stroke. Don't forget the stroke.
Yeah. So I mean, you can't worry about everything.
Yeah. Bless my wife. That's being a hypochondriac.
Awesome. All right, Jeff, there is one more story from the day just before we end up a little bit earlier here that I wanted to focus on, which is we have something from Fractal Health. Fractal Health. We had Jay Kaplan on the show about a month ago, I think, at this point. Very interesting approach, right? Developing a gene therapy to kind of go against GLPs, and it's like activated by appetite. Super cool. So they have popped up on the BioTek Voyager with their Remain 1 data. So up to 84% of GLP-1 induced weight loss retained with Revita versus 46% of that with a sham 1 year of patients receiving it.
So I'm not super familiar with the GLP-1 stuff, to be totally honest. I don't know what the actual benchmarks would be, what the clinical endpoints would be. Like, what is GLP-1 induced weight loss retained with? Is this Revita their therapy versus 46 with sham? Oh, okay, that's okay.
Yeah,
interesting. Duodenal ablations. Okay, huh?
Like 84% weight loss retained, right? Meaning you actually keep it off and you don't have to keep taking a bunch of additional injections, right? This is kind of the whole point of like their GLP-1 gene therapy application, right? You take this once, it goes to your pancreas, you get your insulin activated upon eating, and it kind of helps you keep your weight, and it kind of modulates your
I see. Okay, got it. So it's like, is sham like placebo? Obviously not, because it worked. What is sham? It's got to be some other kind of GLP-1.
No, I don't think so. It's just it is kind of I think it's similar to your placebo. I'll look it up quick.
But it says total body weight change, and they both have reductions here. But then obviously, like, people tend to gain the weight back with like this other form, and this one lasts longer. Because the whole shtick is like it's supposed to be a gene therapy that lasts for a longer time. and you don't got to keep taking the whatever this sham is.
Yeah.
Which is like the goal, right? Like
No,
you don't want to have
a sham trial is a study where some patients undergo a fake medical procedure instead of a real one. It's kind of the same thing as a placebo, but for procedure. Right?
But then explain this to me. How do you get are people just gaining weight? Like sham is nothing, right? So that's weird. I don't know.
Well, there's the placebo effect and just the fact that people are probably going to they're still treating their diabetes and whatnot.
Is it diabetes? I don't know. This looks okay. It doesn't matter. The point is that it lasts for a longer time.
Yes.
It's more durable.
Overall, it is working.
Yes.
Compared to taking something that is just kind of the sham equivalent.
Yeah. Interesting.
Yeah.
Yeah. I mean, what got me with the fractal thing was the method of administration.
Yeah.
Where they go like through the wall of like your stomach and stuff to get to your pancreas, to get it it's like, what? Okay. I mean, if it works, it works.
Yeah.
I think we were talking about how we can use the folks at Sonothera and Fractal Health, like have the Sonothera delivery mechanism be used for Fractal Health's GLP-1 replacement gene therapy.
Right. And you've got the unstoppable gene therapy approach.
Yeah, just like blast your pancreas with some sound waves.
It's not even blast, just kind of like a calm tremor.
Just vibrate them. Give it a massage.
Yeah, we're just gonna vibe the gene therapy in for you.
it's like forget vibe coding. We're gonna do vibe gene therapy.
Yeah, doesn't mean quite the same thing, but we'll run with it. Excellent. Well, should we leave it there, Jeff? Been a great show today. Thank you to everyone who's come. Really exciting to see some of the applications of where cell therapy is going. This is something I've been discussing with a couple different folks recently, right? Like in terms of like the Gartner hype cycle of where cell therapies are. We've kind of been through the peak of disillusionment and then into the trough of despond, and now we're kind of on the slope of enlightenment. And it's really cool to see some of the different approaches that are making some serious traction now.
Yeah. I mean, this is why I built the BioTek Voyager.
Exactly.
To pull these things out of the ether.
Yeah, to map that slope of enlightenment now that BioTek is
such a dork.
You're a dork. We're both dorks. Excellent.
Right. Guilty as charged.
Cool. Excellent. All right. Well, folks, thanks for coming. You have been watching the BioTek Voyager. It's the show that covers what's next in biotech. We're mapping the early stage signals that will be a big deal in a couple years. So thank you for joining us. You are, in fact, early. We'll see you later.